Today we are moving to discuss with our next topic, that is what are sex linkage and what are the sex linked inheritance. Then next topic, again the other topic is disorders, genetic disorders and chromosomal disorders. Okay, so before dealing with genetic disorders and chromosomal disorders, we want to know what are sex linkage and what are the sex linked inheritance. Okay, first one, what is sex linkage? When genes of vegetative or somatic characters that are present on a sex chromosome, okay, that are called sex linked gene. Okay, so when some characters, it may be vegetative character or it may be a somatic vegetative characters, also known as somatic character, that is in besides the sexual characters, that is the vegetative characters, which are present on a sex chromosome. Normally, the sexual characters are present on the sex chromosome, but besides that, some genes that are responsible for some vegetative characters can be seen in sex chromosome. Such type of genes are called sex linked gene, and that phenomena is called sex linkage. Okay, there are two type of sex linkage, X and Y linkage. When it is X linkage, the genes of somatic characters are found on X chromosome. That is called X linkage. The inheritance of X-linked character may be through males and females. It can be either through male or it can be either through female. Okay, that is the inheritance of that somatic character. Inheritance of that particular somatic character which is present on the X chromosome, it can be transmitted to one generation to another through X chromosome, or through males or through female. Okay, so that somatic characters are present on the X chromosome. Example, hemophilia, color blindness. These are the examples of X sex linkage. Okay, then come to Y linkage. It's clear. It is mainly transmitted from one generation to another generation through males only. The somatic character or the vegetative characters, that is the gene which are responsible for a particular somatic characters, are located on Y chromosome. The inheritance of such type of character occur from one generation to another through males only. That's why such type of characters are called hollandric character. Such type of characters are called hollandric character. Hollandric character, hollandric genes means genes which are present on Y chromosome. Hollandric chromosome means simply Y chromosome. Okay, example, these characters are only found in male. So hollandric character or Y linkage, it can be transmitted from one generation to another through males only. So obviously this character which you can see only in the males. Example, hypertrichosis. Hypertrichosis means excessive hair growth in ear pinnae. In ear pinnae, that is a common character which you can find only in males. Then examples of X-sex linkage. Okay, so we know Y linkage is very limited. That is, it can only transmitted from male to male. That is the male character. That is the hollandric genes. Okay, so more inheritance of somatic characters can be found in X-sex linkage. That is, either the character can be sent through male or the character can be sent through female. Okay, so these are the example. Eye color in Drosophila, hemophilia, color blindness, diabetes insipidus. Okay, that is an recessive, autosomal recessive character. Then dystrophy. Okay, muscular dystrophy, that is a autosomal recessive character. Fragile X syndrome, that is also a recessive character. Pseudo rickets, that is a autosomal dominant character. Defective enamel of teeth, that is a dominant character. Defective enamel of teeth. Okay, so this is a fact pattern which is followed by sex-linked genes. First one, autosomal dominant. Okay, autosomal dominant, which means each affected person usually has an affected parent. Okay, which occur in every generation. That should not be what skipped. In every generation, you can see a person who are showing that character. Each affected person usually has an affected parent, which means suppose we people have that particular character, obviously one of our parent have that particular character. That is what we are using, that affected word. Okay, example, Huntington's disease, neurofibromatosis, then acnodroplasia, familial hypercholesterolemia. Okay, familial hypercholesterolemia. So suppose your parents have cholesterol, obviously in your what a mature age, that is 35, 40 plus age, you also may have the chances to cause cholesterol. But actually, that is not 100%, what 100 percentage you may not get the cholesterol. Okay, it's up to your lifestyle. If you are in a controlled manner, you are in a diet manner, and you are taking only good low fatty food, obviously cholesterol will not attack. Okay, so these are the some disease that are autosomal dominant. Then come to autosomal recessive. Okay, so in autosomal dominant means any parent is affected, obviously the children also get the particular character, and it will not skip any generation. Then both parents of an affected persons are carrier. Okay, both parents of an affected persons are carrier. So both the persons should, that is both the parents should can have that character, then only the offsprings also get that particular character. Okay, not typically seen in every generation. It is not necessary to see in every generation, it can be skip the generation. Then sac disease, sickle cell anemia, cystic fibrosis, phenylketonuria. These are the some diseases that are autosomal recessive. Okay, so if you are able to memorize the disease which comes under whether it is autosomal dominant, autosomal recessive, it is more helpful you to attend more questions from this area. Then next one is X-linked dominant. Females are more frequently affected because all daughters and no sons of an affected man will be affected. So X-linked dominant. So females are more frequently affected because all daughters and no sons of an affected man will be affected. Then can have affected males and females in same generation if the mother is affected. Okay, so if the mother is affected, both males as well as females can be produced in affected manner, or those males and females that are produced having the character. Then hypophatemic rickets, that is vitamin resistant rickets. Then ornithine transcarbamylase deficiency. So these are the X-linked dominant inheritance. Then X-linked recessive. Males are more frequently affected. Okay, X-linked recessive means males are more frequently affected. Why? Because females, they have two X. So even one X is affected, it is compensated by the other X chromosome. Okay, that's why males are more frequently affected. Affected male often present in each generation. Okay, so each generation you can see. Example, hemophilia A, then muscular dystrophy. It is the example which comes under X-linked recessive. Okay, based on the sex linkage, there are two type of inheritance. Okay, type of inheritance of sex-linked character: criss-cross inheritance and non-criss-cross inheritance. Criss-cross inheritance and non-criss-cross inheritance. Let's look. Criss-cross inheritance. Okay, so it was found out by Morgan. In criss-cross inheritance, male or female parent transfer a X-linked character. That is, X-linked character means the character which are present on the X chromosome. Okay, the X-linked character to grandson or granddaughter. Okay, so the male or female, they are giving that particular character to their grandson or granddaughter only through offsprings of opposite sex. Okay, through offsprings of opposite sex. If male is male having that particular character, which means male is affected, then their particular daughter, that is the male, will passes that character to the daughter, and daughter will passes to the grandson. Okay, that is a criss-cross inheritance. Inheritance in which characters are inherited from father to daughter and from daughter to grandson, that are called digenic. Okay, so the father, they have the particular character, and the father give that character to the daughter. Okay, then again daughter get married and daughter get a grandson, that is daughter get a son, so that son may have that particular character. So such type of inheritance are called digenic inheritance. That is father giving the character to daughter, daughter giving to the grandson. Okay, then next type, inheritance in which characters are inherited from mother to the son and from son to the granddaughter. Okay, such characters are called diandric character. Mother which passes the character to son, son which passes the character to granddaughter, that are called diandric. Okay, so that is a criss-cross inheritance. Okay, actually father is not giving that character to daughter. That is, daughter is not expressing that character maybe. Okay, so father provide daughter and daughter will passes to the grandson. Okay, so this is a crisscross inheritance. Then, what are non-crisscross inheritance? In this type of inheritance, male or female parent transfer sex-linked character to grandson or granddaughter through offspring of same sex. Okay, in crisscross, you can see the transfer of characters through opposite sex. That is, father giving to daughter, daughter giving to grandson. That is opposite sex. And mother giving to son, son giving to daughter. Okay, that is the opposite sex. Here you can see the same sex. Okay, mother is giving to daughter, daughter is giving to granddaughter, or father is giving to son, son is giving to grandson. Such like that, the character, sex-linked character is transferred from father to the, that is from the parent to the grandsons or granddaughter through same sex. Here that are of two types, that is hologenic and holandric. Okay, hologenic means mother transfer the character to daughter, daughter transfer the character to granddaughter. That is female to female. There is no influence of any maleness. Then holandric. Holandric means father, he is transferring that particular character to son, son is transferring that particular character to grandson. That is male to male. Okay, so if you see the crisscross inheritance, that is the father is transferring to daughter, daughter is transferring to grandson. So you can see a what crisscross manner, father daughter, daughter grandson. That is not a direct manner. Okay, so here in non crisscross you can see a direct without any crisscrossing. They are directly transferring mother to daughter, daughter to granddaughter. Okay, then some other topics, that is sex linked characters. What are sex linked characters and what are sex influenced character? Okay, what is the difference between this? First one, sex limited character. These characters are present in one sex and absent in another sex. That is the sex limited character. Okay, it is only limited to particular sex. Okay, that is not present in both the sex. Either it can be seen in male or it can be seen in female. Okay, but the genes are present in both the sex and their expression is dependent upon the sex chromosome. Okay, clear? The characters are present in one sex only, either female or male. But the genes which are responsible for that particular character can be seen in both sex. Okay, whether that gene is expressed or whether that gene is not expressed, which is depends upon the hormone present on that particular sex. Clear? Sex limited character, that is that character you can see only in either male or either female, but the genes that are responsible for that particular character can be seen in both sex, and that expression of their character, okay, that expression of the gene which depends on the hormone present on the sex. Okay, whether that hormone is present on male or whether the hormone is present on female. Example, secondary sexual character. Secondary sexual character. Okay, genes are located on the autosomes and present in both male and female, but effect of these depend upon the presence or absence of sex chromosome. So actually the secondary sexual character, that is the maleness, all the maleness characters are present in our body also. That is, if I am a female, okay, I am a female, and the maleness character, okay, the genes which are responsible for the production of maleness are present in a female body, but they are not expressing. Why? Because if the genes want to express, particular hormones are required, but our female body will produce only hormones which are responsible for the expression of female sexual characters, not the male sexual characters. Okay, that is the difference. And in male body also, female character genes which are responsible for female characters are also present, but the males are producing only the hormones that produce the or that are helpful for the production of male sexual characters only, male secondary sexual characters only. That's why males are having only male character and females are having only female character. Okay, then next example, genes for beard moustache express their effect only in the presence of male hormone, that is testosterone. Okay, so both male as well as female have the genes which are responsible for beard or the moustache. Okay, both the genes are present, both males and females having the same genes. Okay, but the expression of the genes happen only when the male hormone testosterone is present. Okay, only males will produce testosterone in their particular amount. Females also having the hormone testosterone, but it is on the very low minimum level. Okay, if you keenly observe some females, you can see some females having the moustache or some small hairy appearance on the beard portion, that is the chin portion. You can see some hairs, or in the moustache portion you can see some hairs. That is due to the hormone level, that is the testosterone level is somewhat increased in that particular female, so that obviously the genes responsible for the beard or moustache will express in a limited manner. That's why some little bit of moustache or some little bit of what beard you can see in some females. Okay, clear? Then sex influenced character. What are sex influenced character? Genes of these character are also present on the autosomes, but these are influenced differently in male and female. Okay, in heterozygous condition their effect is different in both sex. So actually genes of the particular characters are present in the hormone, sorry, in the autosomes, but what happened means it is influenced in different. Okay, in male it is influenced in some manner, in female it is influenced in another manner. That is the sex influenced character. Okay, then come to our topic. So these are about the things. So you want to be keep in mind about the sex linked characters, that is sex linkage, what is sex linkage and what are the types and everything. Okay, come to genetic disorder. Okay, so there are two types of disorders are there. Genetic disorders are of two types. Okay, before that, what are genetic disorders? Genetic disorders are disorders, it is caused in an organism or caused in an offspring due to change in genes or chromosome. Okay, so genetic disorders are the disorders due to change in genes or chromosome. Either if a change happened in a gene, it is called Mendelian disorder, and if any change happens in a chromosome, it is called chromosomal disorders. Okay, so the genetic disorder can be of two types. Whether if any changes or modification takes place in a particular gene, then it will be a Mendelian, we will call as Mendelian disorder. And if the disorders happen in a entire chromosome, then we will call as chromosomal disorders. Clear? In under Mendelian disorders, there are different examples are there: hemophilia, sickle cell anemia, color blindness, phenylketonuria, thalassemia, cystic fibrosis. Okay, all these things we are moving to discuss. Then in chromosomal disorders, Down syndrome, Klinefelter syndrome, Turner syndrome, then different ploidies are there. Okay, so we will discuss chromosomal disorders in our next class, but Mendelian disorders we will look today. First one, what are Mendelian disorders? The disorder which caused by alteration or mutation in the single gene. If anything happens in a single gene, okay, anything happens in a single gene and ultimately it results in the change of an expression. It results in the change of an expression is called genetic disorders, that is a Mendelian disorder. Example: hemophilia, color blindness, sickle cell anemia, phenylketonuria, thalassemia, cystic fibrosis, etc. Okay, the pattern of inheritance of Mendelian disorders can be easily traced with the help of pedigree analysis. Okay, last class we already discussed about pedigree analysis. Clear? Let's look. The Mendelian disorders may be dominant or recessive. The Mendelian disorders can be a dominant inheritance or it can be a recessive inheritance. Okay, then the pedigree analysis help us to understand whether the trait is dominant or whether it is recessive. You can see two pictures. Okay, so the pedigree analysis of autosomal dominant trait. Example: myotonic dystrophy. It is an example for autosomal dominant trait. And come to an autosomal recessive trait, that is sickle cell anemia. Okay, we will discuss all these things. So this is the pedigree corresponding to the autosomal dominant trait and autosomal recessive trait. Okay. First one, hemophilia. What are hemophilia? Hemophilia we will call as royal disease. Why it is called as royal disease, I will tell you. Okay, the sex-linked recessive disease. So hemophilia is a sex-linked recessive disease. Okay, if you are able to memorize this, it is more easy to answer the questions. Okay, what hemophilia, which comes under the sex-linked recessive disease, that is X-linked recessive disease. Okay, in this, a protein involved in blood clotting is affected. Okay, hemophilia, a protein which are responsible in the clotting of blood is affected. A simple cut results in non-stop bleeding. Okay, so due to this, if a simple cut or wound is happen in our body, it will continuously bleed, it will not clot. Okay, it is controlled by a pair of allele called capital H and small h. Okay, so the bleeding is controlled by, that is the clotting is controlled by a pair of allele, that is H and H. H is normal allele and small h is responsible for hemophilia. Capital H, it is normal allele, small h is responsible for hemophilia. Okay, then if both the X chromosome having normal allele, then it will be a normal female. Okay, if both the X having normal allele, that is X raised to capital H means normal X chromosome. Okay, then one chromosome having normal allele and another chromosome having hemophilia allele, then it will be a heterozygous female, which means carrier female. Okay, that female is not affected, but that female acts as a carrier. Okay, it can have the ability to pass that hemophilia disease to the next generation. So she may transmit the disease to son. She may transmit the disease to son. Okay, then why it is not transmitted to female? Why? Because in female both are X chromosome. So even though the carrier mother giving affected X chromosome, the father, if the father is a normal person, then the father will give normal X chromosome having the normal allele. So obviously the daughter is become a carrier and she will not affected. Okay, then hemophilic female. So both X chromosome are small h, that is affected, hemophilia affected, then it become hemophilic female. Then come to the male. Okay, come to the male. If X chromosome having normal H, that is the normal allele, then it will become normal female. And if the X chromosome having affected, that is a hemophilia a chromosome, then it will be the hemophilic male. Okay, so these are the conditions. So why it is called as royal disease? Queen Victoria was a carrier of hemophilia. Okay, Queen Victoria was a carrier of hemophilia. So her family pedigree shows many hemophilic descents. So that's why their family may produce so many males, but even though almost all of the males are died due to this hemophilia. Okay, that's why they are ruling females. Females. In females, hemophilia is very rare. Okay, very rare phenomena in females. Why? Because females have two X chromosomes. Even one X chromosome is defect, another normal X chromosome is there to compensate it. So the female will act as a carrier. But it happens only when the mother is at least carrier and father is hemophilic. Okay. So, but it is very rare. Why? Because mother have the ability to become a carrier. Okay, but father, if he is a carrier hemophilic, he will not survive up to a mature age. He will may die up in the before attaining what mature or puberty stage. Okay, any small wound may happen, non-stop bleeding, and results in the death of that particular person. So that's why it is unviable in the later stage of life. Okay, it is not viable. He may die. So this is the pedigree analysis of Queen Victoria. You can see. Okay, Prince Albert and Queen Victoria, they are married and they got Edward. They got Alice, then Edward, Leopold, and Beatrice. Okay, just go through this. Okay, you can see there are so many males are affected, so many females are carrier. Okay, so you can go through it. So these are about hemophilia. That's why it is called as royal disease. Okay, Queen Victoria. Then it has the more possibility that a female become a carrier, not a affected person. Okay, it is very less chances to become a female affected. Why? Because to a female want to affected, then their father and mother should be mother should be a carrier and father should be an affected person. Okay, but that affected father will not be possible because that father will obviously die. A male will obviously die before attaining maturity, those who are a hemophilic male. Then come to color blindness. Okay, color blindness is another Mendelian disorder. It is also a X-linked recessive disorder. So hemophilia and color blindness is a X-linked recessive disorder. Okay, due to the defect of either a red or green cone of eye. So we know our eye have the ability to distinguish different colors. Okay, red color, green, blue, yellow, whatever the color we are seeing, we can easily distinguish the color. Okay, if we are suddenly looking the car, the car is red in color, obviously our eye will read the color, and it passes the information through the connective nerves through the brain, so that obviously our brain will say, okay, it is a red colored car. Like that the eye will do. Okay, so this in color blindness, the disorder is due to defect in either red or green cone of the eye. Okay, green cone of the eye. So it is unable to distinguish whether it is red or whether it is green. Okay, then it results in the failure to discriminate between red and green. Discriminate means to differentiate whether it is red or whether it is green. Okay, so that inability is the color blindness. So it is due to the mutation in some genes in X chromosome. So the color blindness may happen due to mutation in X chromosome. Okay, so yeah, it comes under Mendelian disorder. So obviously the mutation may comes in a nucleotide base pair, only a particular point. It occur in 8% of males. So 8% of the male, if you take 100 people, randomly take 100 people, among that 100 people, 8 people may have color blindness, and 0.4% of female. Okay, why? Because this is due to the genes present in X-linked recessive character. Females have 2 X chromosomes. So even though one X chromosome can have the defect, another X chromosome will compensate. So females will rarely affect. Okay, rarely affect. And you can see, if you are a normal, you have a normal vision, you can see the cap and t-shirt and shoe color in this manner. And if you are a color blindness person, you can see the cap color and the t-shirt color and the shoe color like this. So they cannot differentiate. Okay, in normal person they can see a red color shoe, but an affected person, that is color blindness person, can see a shoe in a light greenish color. Okay, that is the problem. And another, you have a test. Can you read the number in this figure? If no, you may have blindness. I can read. It is okay. I know I can read. You just check by yourself whether you can read the number in that column. Okay, in that circle. If you can, you don't have any color blindness problem. Or if you are not able to read, then you have some problem. You can use this technique to check your parents, grandparents, relatives, and everyone. Okay. Then what happens in color blindness? Normal allele is dominancy. Okay, normal allele is the capital C, and the recessive allele is the small letter C, causes the color blindness. Okay, so normal allele is dominancy and recessive allele is small letter C. Just like we take capital H for normal person, small h for hemophilia person. Okay, the son of a heterozygous woman, the son, that is a carrier woman. Okay, heterozygous woman means carrier woman, has a 50% chance of being colorblind. Okay, you just try to make the crosses. Okay, heterozygous woman crosses with the father. So they have a 50% chances to get color blindness. Okay, a daughter will be colorblind only when her mother is at least a carrier and her father is colorblind. Okay, so a daughter can also get color blindness when what the condition is, the mother should be a carrier, that is heterozygous, and the father should be a affected person. Then only the daughter can have colorblind problem. Okay, so here you can see a carrier woman is there and a normal man is there. Carrier woman marries a normal man, and that carrier woman and normal man produces 4 That is 4 children they produce. In that 4 children, 2 become normal and 1 become colorblind and 1 daughter will become carrier. It is easy. You just put the clues. You can enough produce like this. Okay. So 1 by 4. Then a carrier woman and a colorblind woman. Carrier woman and a colorblind woman. A produce 1 carrier female, 1 affected female, 1 normal man and 1 affected man. Okay. So try to put the crows and just check. So that's all about color blindness. Okay. Then come to sickle cell anemia. So this is the autosomal linked recessive trait. So both hemophilia and color blindness is a sex linked, that is X linked recessive trait. Okay. But here sickle cell anemia is a autosomal linked recessive trait. Okay. It can be transmitted from parents to the offspring when both the parents are carrier for the gene. So what is the condition? It can be transmitted from parents to the offsprings when both the parents are carrier, that is heterozygous for this gene. Okay. The disease is controlled by a pair of allele, that is hemoglobin A, that is normal hemoglobin, and hemoglobin S, that is sickle cell hemoglobin. Okay. HbA and HbS. So homozygous dominant means HbA, that is the normal person. Okay. Heterozygous, that is the carrier one. That is HbA, HbS. It can be a carrier or sickle cell trait. Okay, which means it is a carrier for the sickle cell character. Then homozygous recessive, HbS, HbS. So it will be obviously an affected. That is the autosomal recessive trait. So when the character comes in a recessive manner, then that person will be affected person. Okay, so this is the condition. When the HbA in the homozygous dominant state, it will be a normal, and when it comes in a heterozygous condition, it will be a carrier. When it comes in the recessive condition, it will be a affected person. Okay, so you know the structure of RBC. Okay, it is somewhat a concave disc shaped structure which get the structure to easily pass through the blood vessels. Okay, that RBC get that concave or biconcave disc-like structure, spherical disc-like structure to easily move through the blood vessels without any obstructions. Okay, so what happened to that RBC? Let's look. So actually this has happened. The defect is caused by the substitution. Actually this defect is mainly by some substitutions in the amino acid. Okay, so the glutamic acid is substituted by valine. So we know the genes are responsible for particular character. Okay, how the genes are controlling the character? The genes will undergo. Okay, when you are studying the molecular inheritance of in molecular inheritance, you will understand. Okay, so the genes will produce certain proteins by undergoing replication, transcription, translation. So many processes are there. Okay, so as a result of this translation, as a result of the translation, a protein will produce. Okay, so what are proteins? Proteins are the building blocks of amino acid. Proteins are the building blocks of amino acid. Okay, so many amino acids are joined in a chain manner. So many amino acids are joined in a chain manner, just like this. So many amino acids are joined in a chain manner so that that particular protein will develop. That protein will control the character. If that particular protein produced in our body, that particular character will happen. Okay, suppose a character controls our hair color and that particular protein produced in our body, our hair has that particular color. If that protein is not produced in our body, then obviously our hair don't have that particular color. Okay, so that is why we are saying genes are responsible for a character. So genes will obviously produce a protein and that protein will control the character. Okay, so here what happens in the protein? In the protein, instead of glutamic acid, okay, in the 6th position, you can see first is valine, histidine, leucine, threonine, proline, then glutamine. So in the 6th position, the glutamine is changed to valine. So the 6th position amino acid glutamine is changed to valine. Okay, so the beta globin chain of hemoglobin. So we know hemoglobin is a protein, is made up of alpha helix and beta globin structure. And the beta globin protein structure, it has the 6th chromosome, 6th position glutamine. In the beta globin of hemoglobin, The sixth position is normally glutamine, but it is changed to valine. Then what happened? This single base pair substitution. Okay, this is the base pair substitution mutation. Single base pair change. Okay, single base pair is changed. That is the single base pair substitution at the sixth codon of beta globulin gene. So how in the mutation time I already discussed, each amino acid is caught by a 3 nucleotide pair. That is the codon. Okay, so the AG codon. G, that is what? Guanosine, adenine, guanosine. That codon is changed to GUG. Okay, so GAG codon is responsible for the production of glutamic acid. GAG is responsible for the production of glutamic acid, but it is changed to GUG, which are responsible for another amino acid. Okay, that is valine. So what type of mutation will be there here? It is a what? Missense mutation. Okay, missense mutation means one codon responsible for one amino acid is changed to another codon which are responsible for the production of another amino acid. So here the amino acid glutamine is produced by the codon GAG. So that codon is changed to GUG, which are responsible for the production of another amino acid, that is valine. So this is a base pair substitution mutation, that is the missense mutation. So due to the missense mutation, what happened is the sixth position of the glutamine is changed to valine. Okay, so if the sixth position is glutamine, then that particular person is a normal person. If the sixth position is changed to valine, then that particular person will become affected person. Okay, so then what happened? The normal hemoglobin will become sickled hemoglobin. Normal hemoglobin become sickled hemoglobin. Okay, so the mutant hemoglobin molecule undergo polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc shaped to elongated sickle shaped structure. So you can see this is a biconcave disc shaped structure, and due to this missense mutation, it will become a elongated needle shaped sickle like structure. Okay, sickle like structure. So what happened means, in this structure the blood can easily flow through the blood vessels, but when the blood is converted to, that is the shape of the RBC is converted to another structure, that is sickle structure, it has the ability to bite the blood vessel. Okay, to hit the blood vessel and to accumulate in particular area. Okay, because the blood vessel is mainly developed according to the RBC shape. Okay, so here the RBC shape is changed, so obviously it will causes blockage. So as a result, many problems will develop. Okay, so these are about sickle cell anemia. Okay, in our Kerala, there is a particular place called Attapadi. Okay, in Kerala, that is in my hometown Palakkad, there is a particular place called Attapadi. It is a tribal area. In that area, majority of the ladies and boys, they have sickle cell anemia. Okay, sickle cell anemia. It is what hereditary. They are why? Because why that area is contain the sickle cell means they are tribal people. Okay, they are tribal people. They are not allowed to marry from another area. Okay, only that particular village. Inside the village, they can only marry. Okay, a woman want to marry a man which is present in their village only. They cannot marry a person who is from town area or another tribal village. That's why that diseases is still in that village. Why? Because there is no no variation in the genes. The genes are regulating in that particular village itself. There is no formation or there is no variation in any genes. Okay, that's why that disease is still there. Okay, government put so many effort to reduce the sickled anemia disease, but they are not willing to get out of the village and ready to marry new persons. Okay, they are restricted. Why? Because they are following so many tribal customs and rituals. That's why. Then phenylketonuria. Next one is phenylketonuria. Okay, so actually this is an inborn error of metabolism. This is an inborn error. Okay, it is autosomal recessive trait. It is an autosomal recessive trait. How it is happened? It is due to the mutation in the gene that code for the enzyme phenylalanine hydroxylase. So the mutation is happened in a gene. That gene is responsible for the formation of an enzyme called phenylalanine hydroxylase. Okay, so actually this enzyme converts an amino acid phenylalanine into tyrosine. Okay, so what is happening? This is an inborn metabolism and this is an autosomal recessive trait. So what happened means a gene is mutating here. A gene is mutating here, but that gene is actually responsible for the production of a enzyme. Enzymes are proteins. Okay, enzymes are proteins. Then that gene is responsible for the production of an enzyme called phenylalanine hydroxylase. Phenylalanine hydroxylase. Okay, so what is the role of this enzyme? This enzyme will convert phenylalanine into tyrosine. Phenylalanine into tyrosine. Okay, that is the action of this enzyme. So this enzyme producing genes are mutating. So what happened? That enzymes will not produce. So as a result, the phenylalanine, that is the amino acid phenylalanine, not converted to amino acid tyrosine. Okay. The affected individual lack this enzyme. Due to the mutation happens in the gene, the affected individuals, they lack this enzyme. As a result, the phenylalanine accumulated in our body. The phenylalanine accumulated in our body and it is converted into phenylpyruvic acid and other derivatives. Okay, pyruvate acid and other derivatives. So in a normal person, if we had something, that is our dietary protein, if we had something in that food, if any phenylalanine is there, the phenylalanine hydroxylase that convert the phenylalanine into tyrosine. Okay, then the tyrosine will become melanin, that is the protein which is responsible for the color of our skin and everything. Then other proteins and other dopamine. Okay, dopamine is the signals which are essential for our body. Then if it is a phenylketonuria person that is affected with a gene, then the phenylalanine will accumulate and it will convert it to phenylpyruvic acid. Phenylpyruvic acid. So what will make the what happens when the accumulation of phenylpyruvic acid in our body? So phenylpyruvic acid and other derivative which accumulates in the brain results in mental retardation. So due to the accumulation of phenylpyruvic acid and other derivatives, what happened? Our mental growth will retard. There are also excreted through urine because of poor absorption by kidney. Okay, so these are also excreted through, it can be in minor excretion may happen. Phenyl pyruvic acid can be excreted from our body through urine. Why? Because kidney don't have the ability to absorb these phenyl pyruvic acid. Okay, then you can see the chart unaffected, that is the carrier father and the carrier mother. If both the father and mother are carrier, obviously 1 by 4 of the chance become unaffected person and 2 by 4 chances to become a carrier and 1 by 4 chances to become an affected. That is called phenylketonuria. Okay, so if you can see, some born child may have a very less or retarded mental growth. Okay, so this is due to phenylketonuria. Okay, then next one is thalassemia. So that is why we are checking regularly. If a pregnant woman is checking regularly, that is scanning images are taking, other tests are doing to find out whether they have any genetic disorders. Okay, any chromosomal disorders or Mendelian disorder to the upcoming newly born babies. If they find out any disorders in the womb itself, so we can easily about that particular child. Why we want to waste that life of a precious life by giving birth to such disordered person? Okay, in foreign countries it is frequently doing, they are finding out whether have any genetic disorders or any other problems in the womb itself, and they are aborting such childs. Okay, then what are thalassemia? It is an autosomal linked recessive trait. Thalassemia is an autosomal recessive trait. Actually, it is a blood disease. Okay. It is transmitted from unaffected carrier, that is heterozygous parent to offspring. So actually it is happening from the heterozygous parent to the offspring. It is due to mutation or deletion. It is due to mutation or deletion. Okay, so it results in reduced synthesis of one of the alpha or beta globulin chains of hemoglobin. So what happened? Due to the deletion or mutation happens, either alpha chain is deleted or reduced, or beta chain is reduced. Okay, reduced in the synthesis. So it forms abnormal hemoglobin and causes anemia. Okay, it causes anemia due to abnormal hemoglobin. Okay, so what is the difference between thalassemia and sickle cell anemia? Thalassemia is a quantitative problem. Quantitative problem means synthesis of few globulin molecules also only. Due to the mutation or deletion, what happened? Either the alpha globulin or the beta globulin. Okay, alpha globulin or beta globulin is synthesized very low. That is the thalassemia. What is sickle cell anemia? It is a qualitative problem. Qualitative problem means the disc shaped biconcave RBC is changed to a sickle shaped. That is the quality. Quality of the RBC is changed. Here the quantity of RBC is changed. Okay, that is the difference. Then, based on the chain affected, whether it is affected to the alpha globulin or whether it is affected to the beta globulin, there are two types of thalassemia. That is alpha thalassemia and beta thalassemia. Okay, first one, what are alpha thalassemia? Here, the production of alpha globulin is affected. Okay, alpha thalassemia means alpha globulin is affected. It is controlled by two closely linked genes, that is HBA1 and HBA2, which are present on the chromosome 16 on each parent. Okay, in each parent, in chromosome number 16, this HBA1 and HBA2, that is the genes which are responsible for this, are present in the chromosome 16. Mutation or deletion happens on one or more of the four genes causes the disease. Okay, so in one chromosome, HBA1 and HBA2 is there, and their homologous chromosome pair also HBA1 and HBA2 is there. Okay, so if any mutation or deletion happens to at least one or more of the four genes, that is HBA1 in both chromosome and HBA2 in both chromosome, that is the four genes. Okay, so this may causes the disease. The more genes affected, the less alpha globulin molecules produced. So if one HBA is affected, either HBA1 or HBA2, any one is affected, one gene is affected, so it is not that much the disease is not that much expressed. If four genes are affected, then completely very less amount of alpha globulin will produce. So which may ultimately causes less number of RBC may present in our blood, so we will become anemic. Okay, so that is the alpha thalassemia. Then come to beta thalassemia. Beta thalassemia means the production of beta globulin is affected. Okay, beta for beta. Beta thalassemia for beta globulin. So it is controlled by a single gene. Okay, it is controlled by a single gene. But in case of alpha thalassemia, it is controlled by two closely related genes. Why it is called as closely linked genes? That are present on the same chromosome, HBA1, HBA2. You can see here HBA1, HBA2. That is why it is called as closely linked gene. We already discussed about what are linked genes, what are unlinked genes. Okay, and they are present on the 16th chromosome. But here the beta thalassemia, it is controlled by a single gene, that is HBB, only a single gene in single chromosome. Okay, and it is present on the chromosome number 11 in each parent. Okay, so the mutation of one or both the genes causes the reduction in the production of beta globulin. So obviously RBC will reduce. So as a result, we will become anemic. Okay, so these are about what? The genetic disorders. Among the genetic disorders, we completed the Mendelian disorders. Okay, next class we will discuss about what are chromosomal disorders, and by that we are winding up these principles of inheritance.